In the last video, we focused on what happens when we add a strong acid to catalyze ester hydrolysis. However, the reaction is also accelerated when we add small amounts of a weak acid, and what we find here is that the identity of the weak acid does affect the kinetics. The stronger this acid, the faster the reaction goes. This suggests a fundamentally different mechanism for what's called general acid catalysis, and this is exactly what we observe. The hallmark of general catalysis is partial proton transfer in the rate or turnover limiting elementary step. And what this means is that the business and proton transfer take place at the same time. So in a general acid catalyzed ester hydrolysis, nucleophilic addition and protonation of the electrophile occur at the same time. I do want to emphasize that the role of the acid is still to activate the electrophile. So we still see electron flow from the electrophile to the acid. The difference in general acid catalysis is that at the same time, the nucleophile also engages with the electrophile. So in general acid catalyzed ester hydrolysis, the nucleophile, which here is water, is forming a bond to the carbonyl carbon as the CO pi bond is being donated to HA, the general acid. Using our elementary step formalism, we would label this as an AD sub N for the nucleophilic addition of water to the polarized CO pi bond, plus PT, since the electron flow from the electron sink of the AD sub N step is going toward a proton transfer. These electrons in the CO pi bond are being donated to HA. The intermediate that results is a positively charged intermediate in which the nucleophile has formed a bond to the electrophilic carbon, here the carbonyl carbon, and the carbonyl oxygen has been protonated. So we avoided the protonated carbonyl intermediate that we saw in the last video. We went straight to this protonated alkoxonium, we might say, or this protonated alcohol intermediate, without the intervention of that relatively high energy protonated carbonyl compound. We also generated the conjugate base of the acid catalyst in this step, and this can deprotonate the positively charged intermediate to generate a neutral intermediate. And in fact, this is the exact same intermediate that we saw in the specific acid-catalyzed hydrolysis of esters in the last video. So general acid catalysis is similar to specific acid catalysis in that there's a proton on and business step and then a proton off to kind of turn over or restart the cycle. The difference in general catalysis is that the proton transfer and the business happen at the same time. Another way to put that, a little more formal way, is that partial proton transfer has occurred in the rate-determining transition state. So the slow step, or the rate-determining or turnover-determining step, is right here, and partial proton transfer has occurred in the transition state for this step. In the second half of the mechanism, the general acid catalyzes beta-elimination of the alcohol. Recall in the last video that we protonated the OCH3 group before eliminating it in the specific mechanism. But in the general mechanism, we protonate as the beta elimination step takes place. Proton transfer and the business occur in the same elementary step. That said, the purpose of the acid is still to activate the electrophile or electron acceptor. Here, the nucleofuge is engaging with the acid. It's picking up a proton to ensure that it remains neutral throughout this step. The products of this step are the protonated carboxylic acid, an intermediate we actually also saw in the specific acid-catalyzed mechanism, the conjugate base of the general acid catalyst, and the alcohol, HOCH3. Notice that the alkoxy group, the OCH3 group, remains neutral throughout the beta elimination step. That was thanks to proton transfer from the general acid happening at the same time as beta elimination. To complete the catalytic cycle, we transfer a proton from the protonated carboxylic acid back to the conjugate base of the general acid catalyst. This generates the neutral carboxylic acid and regenerates the catalyst, which can go on to engage with another molecule of substrate and restart the cycle. So now let's contrast general acid catalysis with specific acid catalysis. General acid catalysis is typical of weak acids, which can't, for example, protonate a carbonyl oxygen outright. The thermodynamics just don't make sense. But as water, for example, is attacking the carbonyl carbon, the CO pi electrons can be donated to a weak acid as that step occurs. And the hallmark of general acid catalysis with a weak acid is that proton transfer and the business step, whether it's nucleophilic addition, as in step 1, or beta-elimination, as in step 3, happen at the same time. Proton transfer and the business happen concurrently. In the specific acid-catalyzed mechanism, we have intermediates involving full-on protonation, but we avoid those in the general acid-catalyzed mechanism. Because the acid is directly involved in the business step, the identity and concentration of the acid are very important to the kinetics of the reaction. This is why the reaction is called general acid catalysis. Really, any acid with a variety of different identities for this A group could be used to catalyze this reaction via this mechanism, but the strength of HA, its intrinsic strength, and its concentration both have an impact on the kinetics. Basically, the right way to think about general acid catalysis is that HA is a general structure of an acid. In specific acid catalysis, there is a specific molecular structure, H3O+, in ester hydrolysis, that acts as the acid catalyst.